The overarching question that we work on is, how does a plant survive in the real world? And when you think about it, that's actually a very interesting and complicated one, because a plant germinates from a seed in a particular location and then has to live with that germination decision and grow into a machine that allows the plant to photosynthesize, produce offspring, carry its genome forward in time, and therefore have Darwinian fitness. Because the particular location that a plant has to grow in differs enormously. There's going to be different microbes in the soil, different herbivores, different pathogens, different pollinators, different nutrients, different sunlight levels, different levels of competitors. Because of all that context dependency on growth, plants have had to evolve some very sophisticated signal transduction systems that allow them to perceive the outside world, get signals, and then modify their growth and morphology, and most importantly, their beautiful ability to produce chemicals that allow them to solve all of their ecological problems. So this means that plants that have one particular genome can produce many different phenotypes depending on the exact environment that it locates itself in. And our overarching question is, how does that happen? For the past 20 years, we've been trying to answer that question by querying plants, asking them transcriptionally, what kind of messenger RNAs do you make in a given ecological circumstance, like when a caterpillar eats you? And what kind of small molecules do you make? And what kind of proteins do you make? And what kind of hormones do you make? And then by looking at the answers, we try then to manipulate those responses using RNAi and silencing protein-coding genes. And with those gene-silenced plants that very frequently don't have a phenotype in the glasshouse, we take them out into a nature preserve where the plant actually evolved and has all of the environmental influences that have sculpted that plant's genome for the past millions of years. And then we ask, what's different? And one of the major accomplishments of the past 20 years of research is realizing how important this field work is in the phenotyping process. So after 20 years, we finally have the ability to enter a new phase in our research. A 5-year project of sequencing and assembling the very large genome of Nicotiana attenuata was completed this year, and we published the genome. The genome is enormous. It includes 2.5 gigabases of DNA. And that means that in addition to the 35,000 protein coding sequences that we've been looking at and trying to understand how those protein coding genes have been manipulating and achieving the plant's ability to give you all those different phenotypes from a particular genome, we now have the ability to look at the possibility that many other parts that are not translated into proteins but transcribed into RNA are involved in this regulatory process, in this process that involves both genetic and epigenetic means of shaping a particular phenotype from a given genome. And that is the non-coding RNA. With the availability of a full genome sequence of Nicotiana attenuata, we are now able to look at other ways of regulating a complex phenotype in a more nuanced way. And one of the things we observed when phenotyping plants in the field was that the distribution of small RNAs, particularly the small 22 to 24 mers, were changing dramatically in response to various types of environmental conditions. For example, when an herbivore attacked a plant, there would be a shift of thousands of these small RNAs in their distribution in the plant, and those shifts in those distributions would change throughout the plant. These small RNAs, in conjunction with the very complex phytohormone dynamics, led to the hypothesis that perhaps the important environmentally relevant ways of regulating the phenotype were not through the major protein coding genes, but rather through a more complex and nuanced manipulations of phytohormones and small RNAs to change the phenotype. So with the assembled genome, we could then go after the small RNA machinery. Now, in the Nicotiana attenuata genome, there are 3 RDRs, RNA-dependent RNA polymerases, and these are polymerases that make a double-stranded copy of a single-stranded RNA, and that double-stranded copy of the long piece there feeds into the dicers, which cleave those long strands into 22 to 24 more lengths with a 2 base pair overhang. Those cleaved segments are then fed into another protein machinery called the argonauts, and the argonauts take one of the strands and then use that as a template to find a message RNA which matches that particular strand and then cleaves it. And this way, message RNA, which was produced and matured and sent out from the nucleus into the cytoplasm to make a protein, can be abrogated, can be stopped, so that the phenotype can be adjusted to a particular environmental situation. Now, the genome of attenuata has, as I said, 3 RDRs, 4 dicers, and 11 argonauts. And one of the tools that we've been building over the past many years is to silence each one of them individually by RNAi constructs. That's been successful for all 3 of the RDRs, 3 of the 4 dicers, and 10 of the 11 argonauts. So these are now a toolbox that we can then put together in different combinations, different binary combinations, because they can be crossed together, and the RNAi works perfectly well in a hemizygous state as well as in the homozygous state. So we can silence different components of the toolbox. And we are now ready to be able to manipulate the expression of these thousands of small RNAs and to be able to phenotype the plants that have those small RNA populations altered and constrained in particular ways. And with this, we anticipate, by putting these out into the field and characterizing the small RNA transcriptome profiles, the ability to be able to associate phenotypic change with this more subtle way of regulating transcription, translation, and the like. Why did we think that manipulating the small RNA toolbox would actually be ecologically informative? As I mentioned, that the machinery has diversified in the genome of attenuata, and that's true for all higher plants. There's many copies of all these different parts of the RNA machinery. And as we were developing these lines, the RDRs in particular, 10 years ago, when we put them out into the field, we noticed that when you silence RNA-dependent RNA polymerase number 1, RDR number 1, the plants became very susceptible to herbivore attack. When you silence RDR number 2, the plants became very susceptible to UVB radiation. When you silence RDR number 3, the plants became very susceptible to competitive interactions with other plants. They weren't able to outcompete and grow them. And that already gave us, 10 years ago, the first hint that perhaps the RNA machinery is specialized to particular ecologic functions. And then as we developed the RNAi lines for the dicers, a very similar story emerged from our laboratory screens. The dicer 3 and 4, for example, was particularly specialized to herbivore attack. And then, as we've just published, when we've silenced all 10 of the 11 argonauts, we again see specialization in the argonaut function as well. Argonaut number 8, for example, is a protein that when you silence, the plants become very susceptible to herbivore attack. So overall, one of the key emergent findings of this work is that the machinery that makes these small RNAs is specialized for ecological functions, and that RDR1 in combination with dicer 3 and 4 in combination with argonaut 8 creates a complex of small RNAs that regulate the very complex responses that plants use to deal with things that eat them as herbivores. And we've also found other associations with pathogens and other associations with UVB and other associations with competitive abilities. So one of the things that we really anticipate and look forward to in the upcoming field seasons is examining the phenotypes of these manipulated plants in the field in different combinations. And the way we plan to do that, again, demonstrates the value of field work and how we plan to do it. So, for example, for the herbivore relevant small RNAs, we plan to be planning them out in many populations, hundreds of many populations that include one wild-type plant that's fully resistant, one super susceptible plant that has a major gene in the jasmine cascade silenced so that it's attracting herbivores because it's so vulnerable, and then 2 of the small RNA manipulated lines in different combinations in that micropopulation. So each one of these micropopulations would have a very attractive plant and very vulnerable, a very resistant plant, and then 2 plants that are unknown that we want to phenotype, that have their small RNA machinery manipulated. So the hope is that by doing a lot of sequencing of small RNAs and manipulating the ability to produce those small RNAs in different ways, and then phenotyping these plants in the complex environment in which they evolved with all the different stressors, we hope to be able to make sense and establish causal linkages between the hundreds of small RNAs that are changing at a given time in response to environmental stress. And note that this approach is really fundamentally different from the reductionist approach that tries to narrow down a single microRNA to a single protein target to a particular phenotype that it regulates. We think that this approach has advantages because these signaling systems are evolved in a multi-chaotic, signal-intensive context where a lot of their functionality has to do with crosstalk and phenotype manipulations under many signal input conditions. A point I'd like to make about the discovery that the small RNA machinery is functionally specialized has to do with the value of doing field work and the value of taking plants out into a complex, multidimensional environment and having researchers who are trained in natural history observation to be able to understand phenotypes. We never would have understood that the small RNA machinery was functionally specialized if we had done the classical reductionist approach of isolating individual selection parameters. We simply wouldn't have been able to have discerned that RDR1 plants were unable to ward off predators and herbivores, while RDR2 plants were not able to deal with UVB radiation had we not brought those plants out into the field where they were exposed to this gauntlet of different types of stresses happening all simultaneously. So the same thing holds for understanding this plethora of small RNAs that are changing and being dynamically regulated during various types of ecological processes. That would be almost impossible to dissect in the laboratory setting when you're trying to hold things constant. And the problem with the laboratory setting is that it generates all these other artifacts that we as non-plant observers don't appreciate are artifacts and generating chaotic signals that the plant can't make sense of. So this approach of parsing out the small RNA machinery into various functional units, being able to describe and characterize what happens with the small RNAnome as it changes, and exposing those plants that are constrained in their small RNA production to different types of environmental stresses will hopefully allow us to be able to put the big picture back together again and understand this very complex puzzle of how adaptation occurs. Now, it might be that small RNAs are not really the central players or only a component of the players. And so we have an additional approach up our sleeve that we're planning for the future. And this involves generating another tool that we plan to use that is enabled by the recently available genome. And that is the production of recombinant inbred lines. And we've generated a particular type of recombinant inbred line called the MAGIC recombinant inbred lines. MAGIC stands for the multi-parent advanced intergenerational cross. And what we've done to create this MAGIC line is collected populations of attenuata, seeds from attenuata from populations all over the Great Basin, the entire geographic range of the plant. And then we brought those seeds into the greenhouse, looked for the populations that were most phenotypically diverse, selected 26 populations that we use as the parental generations, and then we generated an intercross by crossing them together, and any gene, any allelic variation that segregates during the crossing over events of sexual recombination can be mapped to a particular chromosomal location in such a recombinant inbred line. And this allows us to impute the function of a small RNA, a long RNA, a protein coding sequence to a particular phenotype. In other words, provide a very unbiased way of understanding the genomic regulators of this phenotypic system. And we hope that by planting out these rills in our field station and being able to do the type of thorough, deep ecological phenotyping that we do on these hundreds of rill lines, we'll be able to learn much more about the genetic regulation of ecological sophistication.